Plants don't just absorb light, they do it with precision. The green leaves we see are complex light harvesting systems designed by nature to capture specific wavelengths of sunlight with remarkable efficiency. This happens through a process known as resonant absorption in their photosystems, which are highly tuned to absorb photons at just the right energies to drive photosynthesis. But why are certain photon energies used and not others? And how do plants achieve such fine control over light capture? The answer lies at the intersection of quantum physics, molecular biology, and evolutionary design. So what is resonant absorption in photosystems? Resonant absorption refers to the idea that a molecule can only absorb light if the energy of the incoming photon exactly matches the energy difference between two of its quantum states. In plants, the molecules responsible for capturing light are specialized pigments like chlorophyll A, chlorophyll B, and carotenoids. Each of these pigments has a distinct energy gap between its ground state and excited state, allowing it to absorb only certain wavelengths of light. Most efficiently in the blue, tilde 430 nm, and red, tilde 660 nm, parts of the spectrum. You might ask, why don't plants absorb all sunlight equally? Because not all light is equally useful. Photons carry energy based on their wavelength. Shorter wavelengths, like blue light, carry more energy, while longer wavelengths, like red light, carry less. If the energy is too low, it won't excite electrons enough to drive the reactions needed for photosynthesis. If the energy is too high, it could damage the pigments or generate unwanted side reactions. So, over billions of years, plants evolved photosystems, called Photosystem 1 and Photosystem 2, that are resonantly tuned to just the right photon energies needed to move electrons along a chain and store energy as chemical fuel. Has this been measured? Yes, scientists have measured the exact absorption spectra of pigments within the photosystems, using tools like UV-vis spectroscopy and femtosecond laser spectroscopy. These studies show that chlorophyll, strongly absorbs at tilde 680 m red light in photosystem 2 and tilde 700 m in photosystem I. These specific energies correspond to the most efficient transfer of energy to the reaction center, where the electron is excited and transferred through the electron transport chain, powering ATP and NADPH production. Why is this important? Because photosynthesis is the foundation of life on Earth, and its efficiency depends on how well plants harvest sunlight. By resonantly absorbing specific photons, plants minimize energy loss, avoid photo damage, and create a robust system that can operate under changing light conditions. In fact, some algae and bacteria have evolved pigments that absorb light in near-infrared or even far-red regions, adapting to low-light environments like deep water or shaded forests. These variations highlight how resonant absorption is tailored to ecological niches. You might wonder, could we mimic this in technology? Absolutely. Artificial photosynthesis systems, solar panels, and bio-inspired energy materials are now being designed using insights from natural photosystems. By choosing materials with energy levels tuned to absorb specific wavelengths, engineers are trying to create solar devices that mimic the selective, efficient light capture found in nature. Some are even exploring how to use quantum coherence and energy funneling, just like in plant antenna complexes, to improve efficiency. So, why do plants harvest specific photon energies? Because only certain photons match the energy transitions in their pigments, and only those transitions can drive the complex molecular machinery of photosynthesis. It's not about absorbing the most light, it's about absorbing the right light. Through resonant absorption, plants have evolved a finely tuned strategy to capture energy from sunlight while protecting themselves and optimizing biochemical yield. Photosystems contain chlorophyll and accessory pigments bound to proteins that fine-tune their absorption spectra through local electrostatic interactions, protein side chains distort pigment conformations, shifting peak absorption wavelengths to precisely match available solar photons. These site-specific perturbations optimize overlap between pigment absorption maxima and the action spectrum of photosynthesis. Altering amino acid environments in engineered variants demonstrably shifts absorption peaks. This molecular tuning maximizes light harvesting efficiency under the variable solar spectrum. A point mutation near the chlorophyll binding site in a light harvesting complex red shifts its absorption maximum by 10 m. It's like adjusting the tint on a car window so it blocks unwanted glare but passes the most useful light. Excited pigments transfer energy via Forster resonance energy transfer, FRET, to their neighbors, funneling excitations toward the reaction center. FRET efficiency depends critically on spectral overlap between donor emission and acceptor absorption. Thus, pigments are arranged to create a downhill energy gradient. This ensures near-unity quantum yield by minimizing backtransfer and dissipative losses. A special organization of pigments within the antenna complex controls transfer rates on femtosecond timescales. Efficient resonance pathways allow even low-energy photons to contribute to charge separation. In purple bacteria, bacteriochlorophyll molecules form concentric rings, where energy hops inward with over 95% efficiency. It's like passing a baton smoothly along a well-rehearsed relay team, where each runner is chosen for perfect timing. Photosystem II and I reaction centers host special chlorophyll pairs with absorption maxima tuned to 680 nm and 700 nm respectively. These specific wavelengths provide just enough energy to drive the endergonic reactions of water splitting and NADP+ reduction, without excess heat loss. The narrow absorption peaks reflect the precise redox potentials required for efficient charge separation. Shifting these peaks too far leads to inefficiencies or photo damage, whereas too little energy cannot drive the necessary photochemistry. Thus, evolution has selected reaction center pigments to resonate optimally with these photon energies. Isolated photosystem II centers lose activity when illuminated at wavelengths longer than 700 dmm, confirming the critical 680 m resonance. It's like tuning a radio exactly to a station frequency. Any slight detuning yields static instead of clear signal. Carotenoids, phycobilins, and chlorophyll variants extend the range of harvestable photons by absorbing in spectral regions where chlorophyll A and B are less active. These pigments pass excitation energy into chlorophyll networks, expanding overall light capture into the blue, green, and orange regions. The diversity of accessory pigments allows organisms to inhabit varied light environments, from underwater depths to shaded forest understories. Each pigment's distinct absorption band is exploited to reduce wasted incident photons. This spectral complementarity increases the total photon utilization efficiency. Red algae use phycobioproteins to harvest green light that penetrates deeper into water, where chlorophyll absorbs poorly. It's like installing multiple satellite dishes, tuned to different channels, so you don't miss any broadcast. Within antenna complexes, low-energy, long-wavelength pigments are positioned closer to the reaction center, creating an energy funnel that directs excitations inward. High-energy pigments at the periphery capture abundant blue light and pass it stepwise through the funnel. This arrangement prevents reverse transfer and minimizes thermal dissipation by ensuring excitations always move downhill in energy. By structuring pigments into concentric energy tiers, plants achieve directional robustness against fluctuations in light intensity. Energy funneling also protects the reaction center from excess high-energy photons. Fluorescence lifetime measurements confirm sequential energy transfer from peripheral chlorophyll B to central chlorophyll A in plant antennae. It's like water cascading down a stepped fountain. Each level ensures flow toward the base without splashing back up. Ultra-fast spectroscopic studies reveal that pigment pairs can maintain quantum coherence, allowing excitations to sample multiple energy pathways simultaneously. This coherent delocalization accelerates energy transfer and improves resilience against static disorder. Coherence lifetimes on the order of 100 femtoseconds enable superposition states that enhance the probability of reaching the reaction center. While coherence decays rapidly, even transient coherence contributes measurably to overall transfer efficiency. Harnessing such quantum effects exemplifies nature's optimization of resonant absorption and energy migration. Two-dimensional electronic spectroscopy of photosynthetic complexes shows coherence fringes between chlorophyll excitons at physiological temperatures. It's like exploring all routes in a maze at once to find the exit fastest, rather than trying one path at a time. Plants adjust their pigment composition and antenna sizes in response to ambient light spectra and intensities, a process known as state transitions. Under low red light, additional light harvesting complexes migrate to photosystem 2, altering absorption cross-sections. High light conditions trigger non-exochemical quenching pigments like zeaxanthin to dissipate excess energy safely. These dynamic reorganizations retune absorption properties to maintain resonant harvesting while avoiding photoinhibition. Such plasticity ensures optimal performance across diurnal and seasonal variations. Shade-grown leaves increase chlorophyll B concentration relative to chlorophyll A, shifting their absorption toward green wavelengths. It's like swapping out different eyeglass lenses for indoor and outdoor lighting to maintain clear vision. Different photosynthetic organisms have evolved distinct pigment suites to exploit specific ecological light niches. Cyanobacteria use chlorophyll D to harvest far red light in deep waters, while desert plants favor pigments absorbing in green gaps. These spectral adaptations reflect the resonant absorption peaks of available sunlight filtered by water, canopy, or mineral substrates. Natural selection tunes pigment-protein interactions to maximize ecological fitness under prevailing light regimes. Comparative genomics of photosystem genes reveals convergent evolution toward similar absorption maxima in disparate lineages. Such diversification underscores the critical role of precise photon energy harvesting in survival. Coral symbionts produce unique fluorescent proteins that shift incident blue light into wavelengths optimally absorbed by dinoflagellate chlorophylls. It's like different radio stations tuning to frequencies that best propagate through urban canyons versus open fields. Researchers emulate resonant absorption principles in synthetic light harvesting assemblies. Dye-sensitized solar cells use molecular dyes whose absorption spectra match chlorophyll resonances. These dyes are bound to semiconductor substrates to channel excited electrons akin to natural reaction centers. Biohybrid systems integrate isolated photosystems onto electrodes to capture and convert specific photon energies with high selectivity. Tuning synthetic pigment protein interactions in vitro enables custom spectral harvesting beyond natural limits. Such biomimetic strategies leverage resonant absorption insights to improve renewable energy capture. A biohybrid electrode coated with photosystem I complexes achieves peak photoresponse at 700 nm, mirroring its natural absorption maximum. It's like installing specialized solar panels designed with dyes that soak up exactly the red and blue wavelengths where sunlight is most intense,